Okay, hi everybody, this is Shane again. I'm excited to introduce you to our last speaker. This is Chani Hodonsky, who is a postdoctoral trainee on the NIH T32 Cardiovascular Research Training Program here at the University of Virginia.
Hi, thank you so much, Shane. Can everyone hear me okay?
Yes.
Great. Okay. So I would like to thank the organizers so much for putting this together on such short notice. I'm excited as an epidemiologist and a geneticist to talk today about inclusive study design at every scale and how that can improve the clinical application of genomic analyses. Let's see if I can. All right. I have no conflicts of interest to disclose. I'd like to encourage people to email me if they have any questions. And this slideshow should be available as a PDF on the wiki as well. So today I'm going to be talking about genomic study design in the context of a disease that we focus on in the Miller Lab, and which is a leading cause of death in the United States that people are probably familiar with, specifically coronary artery disease, or CAD. CAD is the disease that's caused by atherosclerosis, or the buildup of plaques in the arteries that supply blood to the heart, which can unfortunately manifest as a heart attack or myocardial infarction being the first noticeable symptom. Known risk factors for CAD include overweight, smoking, diet, sedentary behavior as opposed to high physical activity, type 2 diabetes, and family history, which of course becomes more important when we begin to think about the genetic context of this disease. But first, a bit of background epidemiology. When it comes to estimating incidence or rates of heart attacks, doing this type of estimation stratified by group can be useful so that differences in incidence due to differences in, for example, access to care can be identified and addressed. This chart shows estimates for white or Caucasian individuals in the state of Virginia. We're really unsurprised to learn that the University of Virginia is located right here. That's where I work. And you can see that according to the legend on the right, the darker shaded counties or cities are areas where a higher number of heart attacks occur each year. Now, when you start to look at race or ethnicity groups that represent a lower overall proportion of the population, incidence can become more difficult to measure accurately, as you need a sufficiently sized denominator or at-risk population to estimate this number precisely. In this chart, we see the same measure as the previous slide, now being reported for African Americans, demonstrating insufficient data for MI incidence or heart attacks in self-reported Blacks or African Americans for not only my city, but 60 other cities or counties in the state of Virginia. And this problem becomes even more pronounced when we start to look at even more underrepresented populations. This is the same chart again for Hispanic and Latinx individuals, which represent nearly 9% of the population of the state of Virginia. However, 130 cities and counties report insufficient data for MI in this population, meaning that if rates of heart attacks do differ by Hispanic Latino ethnicity for a risk factor or disparity that could feasibly be addressed, we have no way of identifying it using this type of measure. Now, there's numerous ways to improve representation of all Americans in incidence rate measures, for instance, but I just wanted to provide this as a background in order to introduce an area of research where we measure risk factors the same way in everyone, which is genomics. So we expect the same genes, although not necessarily the same genetic variants, to play a role in atherosclerosis and CAD and other complex diseases in everyone. So just a quick reminder for people who aren't as familiar with this as I am, DNA refers to the nucleotides that comprise one individual's genetic makeup. The sequence is the same in every cell within one individual person. And a gene refers to a region of the genome that contains information that might be transcribed into RNA and eventually a protein. This is kind of the dogma of genetics that a lot of people are familiar with. And what I'm going to be talking about mostly today is genetic variants, sometimes called SNPs or single nucleotide polymorphisms. That either one or a group of nucleotides at a certain place in the genome might differ across individuals. We can see this if we look at the figure on the right. This represents two people who have the same DNA sequence along the two little strands coming out of conveniently their hearts, until we get to the highlighted area right here, which is a SNP where one person has a G at that location and the other person has an A. It's important to note that genotype refers to the combination of alleles or the reference SNP that one person has at the chromosomes they inherited from their biological mother and father. So for instance, someone in this scenario could be GG, GA, or AA. Several genome-wide association studies have been performed to date in coronary artery disease. GWAS, or genome-wide association studies, typically use some form of linear regression and assume an additive genetic model. And the intent of studies like this is typically to identify genes near the variants that are associated that might play a role in disease and could be good pharmaceutical targets in the long term, or in the short term could be used to explain the pathophysiological processes underlying, in this case, atherosclerosis. In this figure, which is called a phenogram, each blue circle represents one statistically significant genetic variant association with CAD that was identified in GWAS, and the vertical striped bars just represent the 22 autosomal and X and Y sex chromosomes. As one can see, based on the sheer number of associations, CAD is truly a complex disease with over 150 published associations. However, as with other complex diseases, GWAS of CAD have also been performed in majority or exclusively European ancestry populations to date. Eurocentric genomics has long been an issue. So as early as 2011, only 5 years after the first genome-wide association study was published, Carlos Bustamante et al. Published this editorial in Nature calling for increased representation in all types of genomic studies. And they pointed out that findings from GWAS can't be equitably applied unless the original discovery research is performed in populations that are representative of all humans. So why is study population homogeneity problematic? We all have the same genes. One reason is that the likelihood of two variants being inherited together across populations actually differs by population. This is referred to as linkage disequilibrium, or LD, this likelihood. And the reason it's important is because the SNP that might be the most statistically significant in a genome-wide association study might not actually be the causal SNP. It's typically an LD proxy. However, if that proxy is not an LD with the actual causal SNP in all populations, then follow-up analyses to identify that causal SNP can be hindered. Additionally, the large majority of human genetic variation is actually rare as opposed to common, and common variants are statistically easier to detect than rare variants using traditional approaches. So variants in one common ancestry may be, or variants in one ancestral population may be rare in others. The oldest populations on Earth have had the longest time to accumulate both rare and common variants, meaning that Africans have the most genetic diversity within that overall continental ancestry, whereas European ancestry individuals have the least on average. This is important because tools that were used to measure genetic variation for the first decade of genomic research were developed based on European ancestral variants, which means that other population specific variants were difficult or impossible to detect. The x-axis on this figure on the right shows the number of variants in thousands that are common in the population represented by that specific bar, but rare in all other populations. So each bar represents one ancestry group with participants in the 1,000 Genomes Consortium. For example, the bottom bar represents the Luya from Webuye, Kenya. This group of individuals highlighted in red right here represents the European ancestry populations, and the takeaway from this chart should be that studying only these groups means that we're missing out on the opportunity to detect hundreds of thousands of potential other associations in other ancestries. Furthermore, and I don't have time to go into this in detail today, but inclusive study design has also been shown to improve the number and depth of genetic findings and to achieve relevant statistical power with smaller required sample size than European ancestry-only populations. And when we're talking about genotyping hundreds of thousands or even millions of people, this becomes cost-effective as well. Furthermore, a lot of people may have recently become familiar with the term polygenic risk score, or PRS. These have recently gained widespread attention with potential clinical application for predicting a genetic contribution to an individual's risk for a wide array of heritable diseases. However, research has shown that these scores, nearly all of which are generated in European ancestry populations, are not portable to other populations and would be inappropriate to apply to anyone not of European ancestry in a clinical setting, leaving us with a toolkit that's really restricted to only one population. Okay, so back to coronary artery disease from our detour about Eurocentric genomic analyses. So what do we see when we look at genes near the variants that have already been identified? Each section of this figure represents different physiologic processes, with the genes listed under them having been associated with CAD in a genome-wide association study. At the bottom, however, we see this group of 75 genes with unknown role in disease. but we know that these associations exist. So how do we find out what they're doing? We perform an eQTL study, of course. So an eQTL is just an expression quantitative trait loci, and this term means that we're looking at how a genotype or genetic variant might affect expression of a gene. And that kind of looks like this. So in this example, we have SLC17A1 is the gene that we're interested in, and we have a SNP, I would just named it RS37 for short, that is either a G or T at each chromosome, at each of the 2 chromosomes within an individual. And on the right, we can see that as the genotype increases or changes from GG to GT to TT, the gene expression level increases. And this could be associated with disease, which is why we're interested in it. So this becomes where I talk about our study population finally. My PI, Clint Miller, was able to obtain tissue samples from 200 adults who either received a heart transplant or were intending to donate their heart after death and could not be transplanted. It's approximately 35% female, and about 150 individuals actually ended up being able to pass QC for RNA sequencing as well as genotype data. The ancestral representation of our population was broad, and this is important because we're looking at variants across all ancestries that might affect gene expression in everyone. We lack clinical information on a lot of these patients, but we were able to identify inferred ancestry using their genotype information, and that's what this chart shows. Using the legend on the bottom to represent overall continental ancestry, we see that inferred proportion of our overall study population is broadly representative of the United States, with individuals with either complete or admixed ancestry from all continents being represented. An important consideration that a lot of people are probably already familiar with in this group for inclusively designed genomic analyses is avoiding false positives due to population stratification. This is typically done by doing a principal component analysis or looking at PCs. And we did that, of course, in our study as well. And we see that PCs 1 and 2 show separation of African and both South and East Asian ancestry in this example. The number of PCs that are used in a model, it's important to remember, will vary by study and depend on not only the proportion of variants explained, but whether either a small or even a lone individual is driving one principal component, in which case you would not include it. Our lab's work is ongoing, but this is what one might expect an eQTL analysis visualization to look like of the complete results. This is called a Manhattan plot. Each circle represents the lead genetic variant statistical association with the significance increasing according to the y-axis value with a specific gene. So each gene is represented one time, and then the lead variant for that gene is represented once. Statistical significance line is shown in blue here, and this report by Struntz et al. In liver was actually a mega-analysis of 588 people. I will let the audience guess what ancestries were represented in that group. It was Europeans only. So once this eQTL mapping has been completed, we want to see whether there are actually any variants that line up with previously published GWAS variants. And the reason for this is that it kind of narrows the number of molecular experiments that are required to be done to figure out not only whether there's a causal variant in that region, but also whether the gene is actually affecting or having the biological mechanism that we believe it does. That is typically done on a SNP by SNP basis once we have our most significant gene set of interest, and it kind of looks like this. This is a regional association plot where the x-axis shows the significance, with the right being an increase in previously published GWAS studies, and the left showing significance increasing in eQTL studies. It's important to remember that we're looking at individual genetic variants here, so whether that variant is present in all study populations in terms of significant allele frequency to actually be able to be detected is important. Comparing across studies is also really useful. So not only can statistical power be improved by using replication or meta-analysis, but comparing results to publicly available data can be helpful, particularly if you're not sure whether the tissue that you have is actually representative of the disease mechanism that you'd like to be studying. GTEx is a really great example of this resource. And we can see the figure on the right shows ACTA2, for example, is highly expressed in arteries, these tissues right here. However, GTEx and most other publicly available resources, unfortunately, are also generated primarily in homogeneous populations, specifically European ancestry. So comparing results across studies is important. It's also difficult because, as I mentioned before, the majority of SNPs or variants in the human population are rare. And these are more likely to have a causal or large effect size, be causal and have a large effect size, but are less likely to be globally present and can't be detected in populations, of course, where they do not exist. There's hope. Progress is being made. H3 Africa, Kappa, the PAGE study, which I was really fortunate to work with as part of my dissertation work, TopMed, and the International Common Disease Alliance are all putting forth a lot of effort to make sure the representation is increased in genome-wide association studies. But unfortunately, there's still a long way to go. This chart only goes up to 2016, but I hate to tell you that the proportion of representation has unfortunately remained similar in the last 3 years. It's kind of a lot of people kind of passing the buck to the next person in hopes of increasing inclusion while working with the data that they have. And I think that we really need to work together to increase representation of all ancestries, not only because it's relevant to populations in the United States, but because genomics is, a human field of study. So in summary, ancestrally diverse samples are the best for identifying globally relevant associations. And although evidence has shown that this is the most appropriate design, we unfortunately haven't quite gotten there yet. So everybody get together, and if you're on study sections, please ask questions about diversity and representation in studies that come across your table. I'd like to thank my lab and leave people with a thought about whether we are all as objective as we think we are, published in Nature by Angela Cini on only March 9th, so last week. And I'm happy to take any questions.